like to thank the organizers for giving us the opportunity to present our work on the analysis of intertumor heterogeneity using single-cell RNA sequencing in von Hippel-Lindau disease-related renal cancer. Indeed, in VHL disease, the patients inherit from one of their parents a mutation in the VHL gene and later, at the somatic level, a loss of the chromosome 3p can occur and can be followed by a mutation in a driver gene like PBM1, SET2, BAP1, etc. that induces tumorigenesis, and this leads to the formation of several tumors. Each tumor is a genetically independent clone. The hypothesis of our study is that the fact that they are independent clones implies that this tumor may differ at the level of tumor microenvironment, cell signaling, metabolism, and histology, which may have an impact on the response to systemic agents. Our objective is to characterize the differences and the similarities in different tumors from one VHL patient in order to find markers of therapy response as well as single or combination therapy that could work in all the tumors. In order to do this study, we took the advantage of an extreme clinical case of a VHL patient that was enrolled in the OSR VHL program in Milan. The patient had 21 lesions and has been elected for surgery for nephrectomy of the left kidney. A 3D model was built and was used by the pathologist to annotate each lesion, and they choose for us 5 tumors without hemorrhage or necrosis and a normal parenchyma to perform our study. Here is the experimental design of the study. Each sample was divided in 3 pieces. One piece was immediately dissociated and processed for single-cell RNA-seq. One piece was used for pathology assessment and for further validation of the results using immunohistochemistry, and the third specimen was snap frozen for further analysis of bulk RNA-seq and DNA-seq. We assessed the heterogeneity at the level of histology and size. The pathology was clear cell renal cell carcinoma for all the lesions. The grade was grade 2, except for lesion number 4, which was grade 3. And the lesions were PT1A, except for the lesion 1, which was PT3A, so presenting invasive feature. The size were from 0.5 until 5.5 centimeters. So these lesions were heterogeneous at the levels of size, stage, and grade, and there was not always correlation between the different criteria. In order to characterize the composition of the TMA of each lesion and the main expressed genes in each cell, more than 10,000 cells were analyzed by single-cell RNA sequencing from the normal tissue and the 5 lesions. In the integrated analysis, about 10,000 cells were analyzed together. Here is a UMAP representation of this integration of the samples, colored according to the cluster identity, showing 15 clusters which have been annotating using specific cell markers. You see here one cluster with epithelial and tumor cells together, a cluster of myeloid cells with tumor-associated macrophages, monocyte and dendritic cells, CD8 cells, NK cells, T-cell NK, Treg, myofibroblasts, and 2 clusters of endothelial cells. We quantify the cellular composition that was represented here using a bar plot. We can see on the epsilon axis the different samples, including the normal tissue, and on the x-axis the cell population composing them. As you can see, all the 15 populations were present in all the lesions, but they were heterogeneously distributed across samples. Particularly, L5, 7, 2, and 1 have high amount of CD8, and L4, the most aggressive tumor, has lower level of CD8 and high level of tumor-associated macrophages. In order to find common targets of the cancer cell from all the lesions, we checked as the genes up and down regulated in comparison to the normal part, as it is represented here on a Venn diagram. The lesions have 22 common genes significantly upregulated in comparison to the normal renal parenchyma. These genes are involved in hypoxia, metabolism, immune response, and matrix remodeling. We checked for the expansion of the genes in the curved dataset of sporadic CCLCC tumor samples, and saw that all the genes were also upregulated in the sporadic CCLCC. So there is biology shared by all these tumors and also by sporadic CCLCC. We next assess the expression of the genes corresponding to the different therapeutic approaches available. First, we checked the score of the expression of the HIF2 alpha target signature described in the article of Chen collaborators in the cells from the epithelial tumor cluster, showing that the targets were expressed in all the samples and upregulated in the tumors in comparison to the normal part. The lesions with the highest score was number 7, stage 1A in grade 2, and the one with the lowest score was the grade 3 tumor. So it suggests that there was an activation of the HIF2 alpha signaling in all the tumors, with some differences between the lesions. Next, we assessed the inhibitory checkpoint markers coding genes in the cells of the CD8 clusters from the different lesions in the normal part. We saw that these genes PDCD1, FCL2, LAC3, and CDL4 were expressed in all the lesions, and not in the normal part, but with notable differences at the level of the tumors with higher grade. This one. In order to assess the anti-angiogenesis targets marker genes, we checked their expression in the cells from the endothelial cluster in the different samples. All the cells in the endothelial cluster from the different lesions, but also in the normal tissue, express the angiogenesis targets as FLT1, KDR, FLT4, and FGFR1. We next analyze the enriched pathway in each lesion, comparing the genes in cancer cells in one tumor versus all the other ones, and so that the small grade 2 lesion had the immune response pathway, cell death, migration, angiogenesis, and inflammation upregulated in comparison to the other lesions. In the locally advanced lesion in the PT3A, the interferon pathway and the translation processes were upregulated in comparison to the other lesions, and in the more aggressive tumor, apoptosis, migration, oxidative stress and starvation, and mitochondrial and fatty acid metabolism processes were regulated in comparison to the other lesions. To conclude, we successfully performed single-cell RNA analysis from 5 independent tumors and 1 specimen of normal parenchyma, revealing tumors, stromal, and immune cells. There is heterogeneity in the cell distribution, in particular the lesion 4, which has few CD8 cells and high amount of TAM population cells. The lesions have in common 22 genes significantly upregulated in comparison to normal renal parenchyma that are also upregulated in the sporadic CCLCC. The therapeutical targets such as HIF2 alpha targets, inhibitory checkpoint markers, and anti-angiogenesis target genes are expressed in all the lesions but at different levels. The cancer cells from the different lesions have differences at the level of immune response, mitochondrial metabolism, stress, and migration. So our data shows that even if the cancer cells show strong different pathways, there are different shared targets. I would like to thank the VHL Alliance organizations which supported this project, my boss, Professor Salonia, who was at the origin of the VHL program adventure in Milan, the VHL team, in particular Dr. Larker and the collaborators who work with us on this project, in particular the pathologist Dr. Luciano E. Santangelo and the bioinformatician Julia Scotti. Thank you.
Thank you. Thank you for that wonderful talk. Very insightful. Any questions from the audience? Just raise your hands or type your question in Q&A. No? Yes. Okay. We have a question from Dr. Kim.
Hey, sorry to ask so many questions. That's very nice data. I'm very curious, one advantage of the single cell data is that you can tell tumor intrinsic signaling versus immune cell signaling. And so I'm curious, the pathway analysis you did, was that specifically, for lack of a better word, gated or selected for epithelial cells, or was that all cells that you were looking at?
So we are now starting with epithelial cells. So we're starting with cancer cells and epithelial cells, but we are also focused on the endothelial cells for the immune response. For the anti-angiogenesis response, and the immune cells for the immune therapy. But we will go deeply and to look at all the different type of cells. So especially the tumor, the TAM cells, so the macrophages, and all the specific cells that we find, especially the tumor microenvironment. But now we are starting by the cancer cells.
So the data you showed then is cancer cell specific, tumor cell autonomous, correct? Like the interferon gamma upregulation and.
Yeah, they were autonomous. They shared some biological targets.
Very nice.
There were differences anyway.
Thank you.
Great. Thank you for that, Isaline. Wonderful talk.